Hello students, welcome to NCERT CBSE Solutions by Vedantu. Today we are going to continue with our breathing and exchange. So all of you know that yesterday we did the part 1 of breathing and exchange. Today we are going to continue from where we stopped. Okay, so I hope all of you are enjoying the NCERT line by line, especially with all the zoology chapters. So let's continue with the chapter and try to decode NCERT. So students, yesterday we understood how the exchange is happening, and it is totally based on the partial pressure. Yes or no? We understood that yesterday, that partial pressure is the deciding factor from to understand, or to help that where to where movement is happening. Okay, where to where movement is happening. So here, see, alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissues. Oxygen, carbon dioxide are exchanged in these sites by simple diffusion, mainly based on pressure or concentration gradient. Solubility of the gases as well as the thickness. So yesterday I had discussed with you that there are three main factors which is affecting the exchange of gases. First one is solubility of gas. What is the solubility of gas? I hope all of you know that hemoglobin actually does not like oxygen, which is a very sad thing, right? Because hemoglobin is supposed to bind with oxygen, but hemoglobin's affinity with oxygen is comparatively less when you compare it with carbon dioxide or carbon monoxide. So if I have to say, let's say oxygen, carbon dioxide, okay, and carbon monoxide, oxygen, hemoglobin likes carbon dioxide and has more affinity, that is around 20 to 25 times more affinity than oxygen. And when it comes to carbon monoxide, the affinity from oxygen, if you compare, it will be around 200 to 250 times. So hemoglobin has high affinity, okay? Hemoglobin has high affinity towards your carbon monoxide, and because of which it is very dangerous for our body when we are in a situation where there's a lot of carbon monoxide, maybe burning of some fuel or burning of something happens. That means you have to be careful, because there is high chances that, because see, hemoglobin likes carbon monoxide, and imagine you are in a situation where there's a lot of carbon monoxide. It is normal, understood, no, that the hemoglobin is going to, as soon as you breathe in the carbon monoxide, hemoglobin is going to attract carbon monoxide, telling that I like you more, okay, I don't like oxygen that much. So same thing is going to happen. So it is involved in diffusion, and also important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called as partial pressure, okay, and it is represented as PO2 for oxygen, PCO2 for carbon dioxide. So I told you yesterday, partial pressure is like if you have a mixture, okay, let's say this is a mixture of something, this is a mixture of something. In this entire mixture, individually one gas, how much pressure it is exerting, that is going to be the partial pressure. So we represent partial pressure of oxygen as PO2 and partial pressure of carbon dioxide as PCO2. Okay, partial pressure of these two gases in the atmospheric air and the two sites of diffusion are given below. So we saw the table yesterday and we learned how the diffusion is happening. Now there are some more important points that you need to study. See, okay, one second, I'll just read the last line. Similarly, a gradient is present for carbon dioxide in opposite direction, that is from tissues to blood and blood to alveoli. As the solubility of carbon dioxide is 20 to 25 times higher than oxygen, the amount of carbon dioxide that can diffuse through diffusion membrane per unit difference in partial pressure is much higher. So obviously, since hemoglobin attracts, faster to carbon dioxide, there is also chance that diffusion, carbon dioxide going through the diffusion membrane also will be higher, because one of the factors for gas to pass through the diffusion membrane, okay, is solubility of the gas. So since solubility is around 20 to 25 times higher, okay, 20 to 25 times higher, we can obviously think that this one will diffuse faster. Okay, the second characteristic is what, students? Second thing is the partial pressure of the gas. That is, like yesterday we saw, no, if the partial pressure of oxygen is 95 and in another place partial pressure of oxygen is 45, obviously the gas will move from this region to this region, that is from higher concentration to lower concentration. Correct? Yes or no? Then thickness of the membrane. So as I told you, thickness will be less than 1 mm. Okay, thickness of the membrane is going to be less than 1 mm, because we do not want a membrane that is too thick for the gas to do the exchange. Okay, gas to do the exchange. Chalo. Now, The diffusion membrane is made up of three major layers. Very important. Diffusion membrane, okay, is made up of three major layers, namely the thin squamous epithelium of the alveoli, the endothelium of alveolar capillaries, and the basement membrane, basement substance in between them. Okay, so which are all layers, students? We have three layers. Okay, three layers. First thing is a squamous epithelium layer. Okay, a squamous epithelium of alveoli. I will show you the image. Okay, then you have an endothelium. Endothelium of what? Endothelium of your capillary. Okay, alveolar capillary. That means a capillary that is running just below the alveoli to supply. Obviously, if blood is not there, how will the exchange of gases happen? And between this, you are going to see a basement membrane, which is a non-cellular layer. Okay, non-cellular layer, basement membrane. Understood? So, and the basement substance in between them. So these are the three layers constituting the membrane. Now, however, the total thickness is much less than a millimeter. I told you, mm, 1 mm less. Therefore, all the factors in our body are favorable for diffusion of oxygen from alveoli to the tissue, and that of carbon dioxide from tissues to the alveoli. So, one place, always remember that dissociation should happen. Just excuse me for 1 minute, students. This rhinovirus doesn't seem to leave me anytime soon. Okay, this is a true love that has been there for years now, so it's very disturbing. Yeah, so students, look here, this is the alveolar membrane. Okay, this is the capillary running. So which are the three layers that makes the diffusion membrane? This one, the squamous epithelium of the alveoli, okay, endothelium of the capillary, and between, can you see a small space? This is the basement substance. So we can simply say that the diffusion membrane, okay, diffusion membrane, okay, has two layers, two cellular layer, okay, and one non-cellular layer, NC. Okay, two cellular layers because it is the endothelium. I think I'll write it with a different color. I reduce the size of the pen as well. Okay, so two cellular layers, which is the epithelium. Okay, epithelium, okay, squamous epithelium of your alveoli. Squamous means flat. Okay, this is flat cells. Okay, pavement membrane we tell. And endothelium. Okay, endothelium of capillary. Okay, endothelium of capillary. Then here one non-cellular membrane is going to be the basement membrane. Okay, it's not made up of cellular part, so non-cellular part is going to be the basement membrane. Clear? So two cellular part, that is the epithelium or the squamous epithelium, okay, of the alveoli, and endothelium of the capillary. Now exchange is going to happen here. Here the gases are going to exchange like this. Okay, gases are going to exchange like this, and all of you know that the partial pressure here was 104. Yes, from oxygen, so obviously oxygen will pass. At the same way, students, from the tissues the carbon dioxide is going to come back, so carbon dioxide will be allowed to pass this way. Okay, carbon dioxide will be allowed to pass this way and then go outside. So here you will have carbon dioxide to be 0.3%. so faster diffusion will happen, and these people are going to be less than 1 mm. Okay, less than 1 mm. So this is the diffusion membrane. It's very important. If you do not if you forget this, it's going to be actually very difficult for you to understand the concepts. At the same time, questions, high chances of questions coming from these topic. Okay, high chances of question coming from these topics. So diffusion membrane, Okay, now this diffusion membrane, as I told you, two parts are there. Okay, one cellular part, one two cellular parts and two non-cellular parts. Yes or no? So these parts, students, remember that these parts are helping for diffusion. Okay, helping for diffusion. So these parts are going to be helping for easy movement of oxygen and carbon dioxide. Easy movement of oxygen and carbon dioxide. So this portion, let's say this diffusion membrane is the actual one who is going to facilitate that entire gradient difference. Okay, that entire gradient difference is going to happen because of this membrane. So which is the membrane? There is an alveolar layer, okay, and then there is a blood capillary. Okay, blood capillary. So here you have squamous, okay, here you have endothelium. Okay, endothelium is there, and between basement is there. Done. Okay, done. And also remember the factors. Okay, factors affecting diffusion membrane. Question will come. So 3 factors you remember, as I already wrote: solubility, okay, solubility, partial pressure, and the thickness of the membrane. Okay, SPT, PST, anything you can remember. Now, transport of gases. So here, let me tell you one thing. Students, oxygen and carbon dioxide has a different transport system, because oxygen is required for each of the tissues to do cellular activities. Yes or no? To do cellular respiration is why we are even studying this chapter. Yes or no? To do cellular respiration. Okay. Now, how are they getting transported? Okay, what is the transport of gas? So oxygen, if I have to say, okay, if I have to say oxygen, around 3% of the oxygen is going as dissolved, it's getting dissolved in the plasma. Okay, dissolved in plasma. How much? Around 3%. Okay, then 97% is going with hemoglobin. Now students, you're going to study a graph, but before I teach you the graph, you should understand that how this graph is happening, or why this graph is a sigmoid curve. They will ask you this question. First of all, understand that oxygen, okay, is a nice person. What, ma'am? Nice person? No, not like that. See, oxygen is a nice person because oxygen, let's say this is the hemoglobin. Okay, this car is the hemoglobin. Okay, Mr. Bean's car. Not exactly, but yeah. Okay, this is the car. Okay. Students, hemoglobin is the car. Okay, this is hemoglobin. Hemoglobin has a problem. Hemoglobin does not like oxygen. It likes who? It likes carbon dioxide and carbon monoxide. So it's very difficult for oxygen to come and bind with this hemoglobin. So one oxygen will get into the car, okay, and do something called as positive cooperativity. Okay, positive co-cooperativity. That is, this hemoglobin will combine with this oxygen, first oxygen, and they'll nicely sit and chit chat. Okay, chit chat. After that, the hemoglobin will think, actually oxygen is not that bad. Okay, it's actually a nice person. Then the next oxygen will be waiting in the next stop. 4 stops will be there. Then one more oxygen will be waiting in the next stop. Then one more oxygen will be waiting in the next stop. Like this, hemoglobin is going to accept all the 4 oxygens. So that means cooperativity means what? I got into the car. I will not take your opportunity. I will try to make hemoglobin like us more, or make that affinity more, so that next stop you can get it. So this hemoglobin is a car where it is a 4-seater car and only 4 people can sit. So each, understand, one hemoglobin can bind with 4 oxygens. With 4 oxygens. Okay, this is the rule. No more, no less. See, by the time it binds with the first oxygen, okay, when first oxygen comes and sits in the car, the hemoglobin is going to be 25% saturated. Okay, by the time second oxygen comes, it is going to be 50% saturated. And by the time third oxygen comes, it's going to be 75% saturated. And by the time fourth oxygen comes, it's going to be 100% saturated. That means it will be like, I am done. Okay, I am done chit-chatting. I am done with today's social, conversations. I am not going to take in any more oxygens. So this positive cooperativity of the oxygen, that okay, one person gets in and brings another person in, is making the graph a sigmoid curve. So just remember this. So transport of oxygen, students, 2 things is going to happen. 3% is going to go as dissolved in plasma. 97% is going to combine with hemoglobin to form what? Oxyhemoglobin. Okay, oxyhemoglobin. And this is the combination that our body wants. Okay, oxyhemoglobin. We do not want carbaminohemoglobin or carboxyhemoglobin, but they also happen. Okay, they also happen because of the affinity. So look here, blood is a medium of transport for oxygen as well as carbon dioxide. About 95% of the oxygen is transported by RBCs in the blood. That means simply hemoglobin plus oxygen complex. How much percent? 97%. Okay, 97%. Now the remaining 3% of the oxygen is carried in dissolved state through the plasma. So I told you dissolved in the plasma. Nearly 20 to 25% of carbon dioxide. Now here, when you talk about carbon dioxide, it's a different story by itself. Did I leave a place here? No, wait. Okay. I'll write here, student. This is unnecessary actually. Very small concept, okay? We can finish it soon. Yeah. So when we talk about transport of carbon dioxide, remember that three things is going to happen. Okay, three things are going to happen. Three things are going to be, first, 7% of the carbon dioxide is going to be dissolved. 7%. Okay, then to be precise, okay, if I have to tell you the exact number, it's going to be 23% of carbon dioxide will go as combined with hemoglobin to form carbaminohemoglobin. Okay, carbaminohemoglobin. Okay, carbaminohemoglobin. Then the rest, okay, rest 70% will be stored as bicarbonates. Will be stored as bicarbonates. Understood, all of you? Yes or no? Got it? This is what we are going to discuss. Okay, this is what we are going to discuss. I have to kill rhinovirus, students. I have to make a variant that will kill rhinovirus. I mean, I have to make a antibody that's strong that rhinovirus never affects me. Okay, you know why rhinovirus keep affecting? Like, you can have a question, oh ma'am, how does rhinovirus always affect you? Like, don't you take tablets? It's very simple, because rhinovirus have multiple variants. Okay, rhinovirus always come in different variants. And when we put fan, when we drink cold water, automatically I don't drink cold water, but yeah, fan and AC affects. Okay, so 7% is gone as dissolved, students. Rest 23% exactly will be gone as carbaminohemoglobin and 70% as bicarbonate. So this is how your transport of carbon dioxide happens. Okay, and we'll be learning how bicarbonates will be formed. Okay, now you have to learn a graph. So that's why this introduction is given. Okay, same thing in hemoglobin also. I forgot to tell. Same thing in carbon dioxide also, hemoglobin. But hemoglobin likes carbon dioxide, so there is no positive cooperativity, like please accept us, nothing like that. Hemoglobin plus carbon dioxide, and same thing happens. 4 carbon dioxide will be able to move. Why 4 carbon dioxide? Because this hemoglobin, okay, this hemoglobin, students, this color. Okay, 1 second. Which color to take? I'll take blue, or I'll take something else. Yeah. So students, this hemoglobin, this is hemoglobin, this is hemoglobin chain, this is hemoglobin chain, this is hemoglobin chain. Okay. Now, this hemoglobin chain, there are Fe ions, okay, who is going to help in the binding. Okay, the heme group. Okay, so this binding, each of them can bind with 1%, right? Here also an oxygen, here also an oxygen, here also an oxygen, here also an oxygen. Got it, all of you? So there is a heme group, okay, and a globin group. Heme group and a globin group. Got it? This heme group will have 4 Fe2 plus state. 4 Fe2 plus state. And globin will have 2 alpha chain, 2 beta chains, which you don't have to remember. But yeah, 2 alpha chains, 2 beta chains. Okay, 2 beta chains. So you 2 beta chains. So these groups are going to help in the binding. Now, being said that, you understood that 4 oxygen, 4 carbon dioxide only can sit in the hemoglobin. Okay. Now see here, transport of oxygen. Hemoglobin is a red colored iron containing pigment. I told you heme group as Fe ions, okay, present in RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Okay, each hemoglobin molecule can carry a maximum of 4 molecules of oxygen binding. Oh my god, I'm so sorry. Please excuse me, students. Each hemoglobin molecule can carry a maximum of 4 molecules of oxygen. Binding of oxygen with hemoglobin is primarily related to the partial pressure of oxygen. Okay, partial pressure of carbon dioxide, hydrogen concentration, and temperature are other factors which can interfere with the binding. That means if partial, so sad students, if oxygen needs to bind with hemoglobin, okay, let's say we have other people as a problem. We have to bother about how much carbon dioxide is there, how much H plus is there, what is the temperature. So think of these two people as Nanban. Okay, Nanban means friend, no? Nanban movie, all of you know, three idiots. Okay, if this fellow is increasing, this people also will automatically increase. Like, you know, there's like one friend, no, who does something, he'll jump into well, even you people will jump into well. Like that, these people are. Whatever carbon dioxide does, they will also do. So these factors, if they are all high, then oxygen's, you know, binding with hemoglobin is going to be broken. Okay, it's going to be broken. Because they will not, see, the moment carbon dioxide is more, hemoglobin will be attracted to carbon dioxide. Okay. The moment carbon dioxide is more, then there is chances that oxygen will have to dissociate. Okay, dissociate. What is dissociate and associate, students? See, associate means, this is a big problem now, cold. Oh my god, it's getting worse. So I'll see how much I can take the class, students. Okay, after that I'll stop. As you can see, it's all red. Okay, so students, what is association and dissociation? See, association means, okay, association means when hemoglobin can bind with oxygen, okay, and form oxyhemoglobin. Okay, oxyhemoglobin. Okay, now dissociation means what? He is leaving it. Okay, divorce is happening. So dissociation means hemoglobin, okay, with oxygen complex is going to break and get hemoglobin plus oxygen. Understood? Here breaking is happening, and this breaking will happen when carbon dioxide is more. Okay, when carbon dioxide is more, let's say this is the mother-in-law, this is the father-in-law, this is the sister-in-law. When mother-in-law is there, the husband-wife is going to break. This is an example. Okay, now partial pressure of carbon dioxide, hydrogen ions, and temperature are other factors which can interfere with the binding. A sigmoid curve is obtained when the percentage saturation of hemoglobin with oxygen is plotted against PO2. Okay, that is, as I told you, hemoglobin's saturation is going to be at 4 levels: 25% when 1 binds, 50% when 2 binds, when 75% when 3 binds, and 100% when 4 oxygen comes and binds. So depending on the saturation, we are going to plot a graph. Okay, now this curve is called oxygen dissociation curve. So what is this curve called? Oxygen dissociation curve. Very important curve. Now, as it is highly useful in studying the effects of factors like, see, we'll study about the partial pressure of carbon dioxide, H plus concentration, and temperature, and on binding with oxygen with hemoglobin in alveoli where there is high partial pressure of oxygen. How much partial pressure of oxygen is around 104? Okay, low, how much carbon dioxide is there? Carbon dioxide is around 40. Yes or no? Correct? Yes, perfect. Now, lesser H plus concentration and lower temperature. The factors are all favorable for the formation of oxyhemoglobin. So from here, you can expect that HbO2 will be formed. Okay, why? Because this fellow is high, this fellow is low. If this fellow is low, automatically H plus also will be low, temperature also will be low. Correct. Now, whereas in the tissues means, students, just imagine I'm holding on to something, okay, I'm carrying something and going. This is like my life, okay, I'm carrying this glass box and going. Now you imagine I carried it from the alveoli, okay, this box, okay, I am hemoglobin, this is oxygen, I'm carrying it from the alveoli, I'm going to the tissues. Now what is my job in the tissues? I have to give this oxygen to the tissues so that tissues can do cellular respiration. Now if I hold on to the oxygen, can the tissues happily survive? No. So for to break our bond, okay, obviously after binding, then bond is going to be there. To break our bond, what is going to happen in the tissues? Lot of carbon dioxide will be there, because they would have used up all the oxygen which we gave, and they will be left with only carbon dioxide. So they will take as the carbon dioxide increase, hemoglobin will be like, oh, I have to get attracted to carbon dioxide, yeah, oxygen, it's okay, so it will leave the oxygen. So I will leave the oxygen here in the tissues and I will go, okay, I will go with carbon dioxide. So there dissociation will happen, understood? Association will happen in the alveoli, dissociation will happen in the tissues. Understood the difference? If you understand the difference, there is nothing difficult about this graph, okay? People complicate this graph, it's not required, that much complication is not required in life, okay? So whereas in tissues, low partial pressure of carbon dioxide, high partial pressure, yes or no, in tissues obviously carbon dioxide is going to be there, high temperature concentration, high H plus. I told you whatever carbon dioxide does, these two non-bonds are going to do, okay? The conditions are favorable for dissociation for oxygen from oxyhemoglobin, so we can give the oxygen to the tissues. This clearly indicates that oxygen gets bound to hemoglobin in the lung surface and gets dissociated in the tissues. Every 100 ml of oxygenated blood can carry 5 ml of oxygen to the tissues under normal physiological condition, okay? When a person is doing exercise or other thing, it can go up to 15 ml, but normally it is 5 ml. And there is actually a calculation where you calculate the atrial blood minus the venous blood, where atrial blood is around 19.4 minus 14.4, you will get around 5 ml, okay? We can do this calculation. And for if we have to like calculate for hemoglobin, okay, if you have to calculate for hemoglobin, we'll calculate that 1 gram, no, oxygen, 1.34 ml, okay, of oxygen. So like this we do calculation usually, okay? So for 100 ml, how much it will be? 100 ml, it will be 1.34, okay, into you do, and you get approximately how much, students? So you have to calculate for every 100 ml, there is 15 grams of hemoglobin. That much hemoglobin is there, means only that much oxygen can be carried. So every 100 ml will have 15 grams of hemoglobin. So every 100 ml is equal to 1.34 into 15, because hemoglobin is the one who is carrying the oxygen. So how much is the calculation? 1 second, let me just do it. 1.34 into 15. Okay, around 20.1. okay, so approximately 20.1. okay, 20.1. Now we calculate approximately 20 ml of oxygen will be taken. Then we have something called as atrial system, that is from the heart, okay? Atrial system and venous system. Atrial system is around 19.4, this is around 14.4, and we minus it, we'll get 5 ml. This calculation is there, one more calculation is there, but you don't have to know the calculation, okay? Now look at this, student, this is an oxygen dissociation curve. Now listen to me carefully, okay? Try to understand with all your mind and soul, because this is a little task, okay, to understand it, but it's very easy. It's not like, oh my god, I cannot understand it at all and all of that, nothing like that, okay? Nothing like that. First, rhinovirus is the problem, okay? Okay. Can't do anything, students. Some days are like this, especially in the mornings it is like this, okay? Especially in the mornings it is like this. I think many of you will be facing, okay? Many of my students have actually very extreme cold in the morning. So I have it, yes. So listen to me carefully. The curve, okay? The curve, see, how do I tell you now? The curve, the curve can shift to the right and left, okay? This is to the right, okay? This is to the right, and obviously this is to the left, okay? Left. Now right and left, students, matters, because at the right, okay, if the shifting has to happen to the right, that is when your carbon dioxide is extremely high, okay? If carbon dioxide is high, your H plus is going to be high. If if carbon dioxide is high, temperature is going to be high. I told you, when these three people are high, oxygen has a chance to repel and go, right? Oxygen will be like, I don't want to stay here, I'm going to leave. So oxygen will shift towards the right. That means here dissociation is going to happen. Okay, dissociation is going to happen where the oxygen will move towards the right. Okay, oxygen will move towards the right. Understood? That is, oxygen is going to split up from hemoglobin and become Hb plus O2. Okay, this HbO2 will become H plus O2. Yes or no? Got it? So this is the situation. Now if it is shifting towards the left, okay, if it is shifting towards the left, what is going to happen? That means here the partial pressure of oxygen is going to be high. Okay, and here obviously if this fellow is high, this fellow is going to be less. Okay, H plus also is going to be less. Okay, temperature is going to also be less. Yes, so all these people are going to be left. So this is actually the curve. Okay, dissociation and association curve. This is the whole curve. Here you have percentage of saturation of hemoglobin with oxygen, partial pressure of oxygen. So I'm extremely sorry, I just go. Okay, I'll take a tablet and come. Otherwise I can't stay, I can't teach, I have to finish the chapter also. Okay, just give me a just give me a 10 minutes break. I'll take a tablet and maybe drink some hot water and come. It's getting very worst. Yes, students, I'm back. Okay, I think it's a little bit better. 1 second, audio. Am I audible now? Yes, audible, right? Awesome. Okay, let's continue. Okay, so as I taught you the curve, look at this curve here. So what is exactly going to happen is if the curve has to shift towards the right, okay, if the curve has to shift towards the right, that is when there is reduced affinity with oxygen and hemoglobin. And why does this happen? Obviously because we need to do dissociation and we need to drop the oxygen there for the tissues. See, if there is a bond, okay, we have to break the bond, no, to release something. So that bond is broken so that we can release this oxygen to the tissues and tissues can utilize it. Okay, that time what we can do is to break the bond, we have to increase the carbon dioxide level, and in the tissues carbon dioxide is more. So when automatically carbon dioxide increases, oxygen will decide to move towards the right, make the right step of stepping outside. Okay, so it will dissociate and it will go and bind with the tissues. So this will happen when there is a high carbon dioxide, lower pH. So pH and oxygen is kind of going to be, aligning with each other, because pH also will be less, that time oxygen also will be less, okay, and higher temperature. But carbon dioxide, these three factors, okay, H plus also, temperature also, these three factors affect the dissociation. If these three factors are going to help in the, dissociation or association. Here what is going to happen, students? Here association is going to happen. That is, binding chances are more when the affinity is more. So there will be high affinity of hemoglobin with oxygen because there is low carbon dioxide, higher pH, as I told you, this two will be the same, and lower temperature, and obviously lower H plus concentration. Okay, lower H plus concentration. Okay, now curve is clear, no? Now transport of carbon dioxide. So students, now we are going to study a mechanism of how the bicarbonates are going to be formed. So transport of carbon dioxide. See, carbon dioxide is carried by hemoglobin as carbaminohemoglobin. Okay, as what? Carbaminohemoglobin. Many students write carboxyhemoglobin. Please try to understand carboxyhemoglobin. Carboxyhemoglobin is when carbon monoxide binds with hemoglobin. Okay, carbon monoxide binds with hemoglobin. Yes, not when carbon dioxide binds. Okay, this binding is related to the partial pressure. This binding is related to the partial pressure of carbon dioxide, and oxygen is a major factor which affect the binding. So same thing. If oxygen's binding, we have to know. If oxygen's binding, we have to note it. Then definitely carbon dioxide is going to be the factor. If carbon dioxide we have to note, oxygen will be the factor. They're going to be obviously simultaneously going to help. When carbon dioxide is high and oxygen is low, as in tissues, more binding of carbon dioxide occurs, whereas when ulta happens, more binding of oxygen happens. Okay, now see here what happens. Here, carbon dioxide, which is bound to the hemoglobin from the tissues, is delivered to the alveoli. RBC contains a very high concentration of enzyme called carbonic anhydrase, and minute quantities of the same is present in the plasma too. This question will come to you asking where is carbonic anhydrase present. You have to tell it is mainly high concentration is present in the RBC, very small quantity is present in the plasma. This enzyme facilitates the following reaction. What reaction exactly? In the RBC, carbon dioxide plus water is going to bind. Normally also this will bind, students, but it happens at a very less rate, okay, or very at a very slower rate. To increase, we will use an enzyme called carbonic anhydrase, which is present in the RBC. This will increase the formation of carbonic acid. Okay, this will increase the formation of carbonic acid. Now this carbonic acid breaking also is going to be done by your carbonic anhydrase. Clear? Yes, very good. Okay, breaking also is going to happen because of carbonic anhydrase, and it is going to give us, okay, it's going to give us HCO3 plus H plus. What is it going to give? HCO3 plus H plus. Okay, now this HCO3 will go into the plasma, okay, bind with salts in the plasma like NaCl, and form NaHCO3, which is a bicarbonate. So this bicarbonate is what we were talking about. Okay, this is the bicarbonate we were talking about. That is 70% of the carbon dioxide is transported as bicarbonate. Is this clear or not to all of you? Okay, clear? Yes, clear or not? Okay, 1 second, students, as usual. And always, whenever you are using like whenever you're wiping your nose, always use a softer cloth, or else this layer of the skin will completely rip off, especially if you have cold like me every morning. So it's very dangerous. So always use a soft long cloth. Okay. Now see, at the tissue site where the partial pressure of carbon dioxide is high due to the catabolism, carbon dioxide diffuses into the blood and forms HCO3 and H+. At a alveolar site where, so basically they're telling reverse, what happens in reverse. So look here all of you. Let's, okay, let's, okay. 1 second. Okay, now I'm going to draw an RBC. I think RBC should be bigger. Okay, first we'll draw RBC, student, then we'll draw the what it is, because I'll be able to write the reaction, to write the whole reaction, no? Kind of dumbbell shaped. Okay, this is your RBC. Okay, this is the RBC. This is going to be the plasma. Okay, here also plasma. Now inside the RBC, what they told is going to happen, the carbon dioxide is going to enter into the RBC. Okay, it is going to enter, bind with water, okay, with the help of Ca, that is carbonic anhydrase, form H2CO3. Yes or no? H2CO3. Now this H2CO3 will dissociate again with the help of carbonic anhydrase. It's going to dissociate to form HCO3 minus plus H plus. Now this HCO3, students, it's going to go into the plasma. That is going to be HCO3 is going to combine with the NaCl in the plasma and form NaHCO3 and plus Cl minus. Now all of you look here, this minus negative ion, I have taken it outside RBC. So don't you think there is going to be a difference in the ionization and the difference in the charges? Okay, difference in the charges in the RBC. Yes ma'am, because you took something negative outside, you might have to give something in return. So what I will do, I will give this chlorine in return for this HCO3 to maintain the charges, okay, to maintain the ionization, and this is called as chloride shift. This is called as chloride shift. Understood? Haldane effect we'll teach again, but this is called as chloride shift. Clear or not? Now this is one process. Okay, now if reverse has to happen, what is going to happen? This NaHCO3 is going to break down to HCO3 and NaCl, and this HCO3 is going to enter inside and continue the cycle and go to the alveoli. Okay, go to the alveoli. So this is all it should. This is all. Okay, this is RBC. Okay, 1 second. This is RBC. RBC's color. Which color do we do? Okay, RBC. Yes, okay, this is RBC. Okay. This carbonic anhydrase is going to be present here also, because understand all of this is a very slow reaction. To increase the pace of the reaction, we have to use this. So this is what is going to happen, and this is how the bicarbonates will be formed. Okay, this is how the bicarbonates will be formed, and around 23% of oxygen will combine with hemoglobin again in the RBC itself. Okay, in the RBC to form HbCO2. This also will break while releasing, like when I have to release this carbon dioxide. Okay, when I have to release this carbon dioxide, remember that this carbon dioxide breaking is going to happen from the bicarbonate, like it's going to be a reverse reaction, no? So what is going to happen here? NaHCO3, okay, is going to break and form HCO3 plus NaCl. Okay, this HCO3 is going to enter the RBC. Okay, it's going to enter the RBC, and this HCO3 is going to, student, this H plus, no, which we broke there, okay, to form HCO3. H2CO3. Now remember this H plus, which I did not write here, just remember that this H plus is going to combine with hemoglobin. Okay, it is going to combine with hemoglobin and it is going to act like a buffer. Buffer means what? Excuse me. Okay, buffer means what? Something that will maintain the pH. Okay, something that is going to maintain the pH. Maintain the pH. Okay, something that is going to maintain the pH. So this HB, what this combination also, what is that? HHB is also going to be there. Okay, is also going to be there. Now, what happened? Now students, look here. Here, what is going to happen? This H plus, no, this HHB is going to break and HB plus H plus is going to come and then they are going to combine. Now this will go and this will again break to H2O plus carbon dioxide. Now this carbon dioxide is going to the alveoli and from alveoli it will go to the atmosphere. Okay, alveoli to the atmosphere. Clear or not all this? Just look at this. Okay, by then ma'am will just okay, look at this. Any doubts you can always ask. Okay. Oh my god. Yes. Okay, clear. Okay, clear. I hope this is clear. So what happens from the top? Okay, let's say carbon dioxide plus hemoglobin will form hemoglobin complex. Okay, hemoglobin plus carbon dioxide. That complex also will break when we have to give this carbon dioxide to the alveoli. Okay, first thing happens is carbon dioxide plus water in the presence of carbonic anhydrase, an enzyme which is rich in RBC but less in plasma, will combine to form H2CO3, carbonic acid. That carbonic acid again will split to form H2O plus carbon dioxide. This H2O plus carbon dioxide, students, no, they will form HCO3 plus H plus. HCO3 will go outside, that is it will go to the plasma, and because there is a change in the ions, because one negative ion went outside and RBC is left with no negative ions, definitely there's going to be a change in the ionization. So this HCO3, instead of that HCO3, we will give the chloride ion, telling that you take the chloride ion because we have taken your HCO3 and this is called as chloride shift. This HCO3 will now come outside, combine with the NaCl in the plasma and form NaHCO3, which is a bicarbonate. Now when alveoli is demanding us, telling that give the carbon dioxide because I need to throw the carbon dioxide outside or this person is going to exhale, what is going to happen? This bicarbonate is going to break. Okay, it's going to break. At the same time, the hemoglobin complex, HbCO2 also is going to break, and all of this is going to go to the alveoli. So a reverse reaction happens. Okay, a reverse reaction happens just like this. Okay, just like this. Okay, full thing reverse will happen. This is also called as the Haldane effect. Okay, this is also called as the Haldane effect. Clear? Now this is how the transport of carbon dioxide is there. Now we'll come to the end of this topic. We have just 2 topics left. Now look here. Who is controlling this respiration, or who is controlling the whole of it? That is what you have to see. Okay, human beings have a significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural system, a specialized center present in the medulla region of the brain called respiratory rhythm center. Where is it present? In the medulla region. So whenever we study the function of medulla, we tell it helps in breathing. So that is because of having the RRC, that is the respiratory rhythm center, is primarily responsible for this regulation. Another center present in Pons region of the brain is called as the pneumotactic center, which can moderate the function of the respiratory rhythm center. So this will act as a moderator. So remember, RRC is the main fellow who is going to do the function, and your pneumatic center is going to be the moderator who is going to moderate this fellow. Okay, it's going to moderate the functions of the respiratory rhythm center. Neural signals from this center can reduce the duration of inspiration and thereby alter the respiratory rate. So it can alter how much air we take in. Okay, a chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to carbon dioxide and hydrogen ions. Why, students? Because carbon dioxide and hydrogen ions are necessary to understand if we have more oxygen or less oxygen. When these two people are high, we automatically know that it is time for dissociation because oxygen is less. Okay, increase in the substance can activate the center, and which in turn can signal the rhythm center. So one thing that you need to understand is you are not breathing because there is lack of oxygen. You are breathing because there is more carbon dioxide. Okay, so actually oxygen has hardly any, like what to tell, oxygen has a very like very small role in this whole regulation, which is shocking to all of us because we think that, oh my god, my body needs oxygen, so I start inspiring. No, you take in air or you give out air because your carbon dioxide H plus and your temperature is different, and there are centers who are going to recognize this. Okay, so increase in the substance can activate this in which this centers, which in turn can signal the rhythm center to make necessary adjustments in the respiratory process by which these substance can be eliminated. Receptors associated with aortic arch and carotid artery can also recognize changes in carbon dioxide and H plus concentration and send necessary signal to rhythm centers for remedial action. So the role of oxygen in regulation of the respiratory rhythm center is quite insignificant. Understood? Main job who is going to do, carbon dioxide is going to do. So students, we have who and all we have RRC, okay, respiratory rhythm center, which is present in the medulla. Okay, we have pneumotactic centers. Okay, you have pneumotactic center, which will act as a moderator to help this fellow. Then we have chemosensitive areas, which is going to check the concentration of carbon dioxide and H plus concentration. When these two are high, okay, when these two are high, automatically the centers are going to get activated. Okay, the centers are going to get activated. Then we also have carotid arteries. Okay, carotid arteries and aortic arches, okay, which is also going to regulate depending on the concentration, okay, concentration of H plus ions. Okay, concentration of H plus ions. So this will let us know that if who is more. Okay, carotid and aortic arch can also recognize the changes in the concentration of carbon dioxide and H plus, okay, and send necessary signal to the rhythm center telling that you inspire air or you, depending on what you want to do, okay, depending on the body's condition. Now, disorders of respiratory system, students, very simple disorders which you have learned in your previous classes as well. Okay, disorders of your respiratory rhythm center. Oops, bad day. Okay, asthma is a difficulty in breathing causing wheezing due to inflammation of the bronchi and the bronchioles. So all of you remember, no, like this, your bronchioles are there. So when there's swelling happens, okay, when here swelling happens, this will cause asthma, because of which lack of oxygen will be there, and this will cause us wheezing. We will try very hard to get in the air. Okay, next is emphysema, which I have taught you in biotechnology. We need alpha-1 antitrypsin, which will there in our body, which is a human protein that actually cures, or let's say gets us away from this emphysema. But as we smoke continuously, this will completely vanish, and then we'll have to use a transgenic animal to get this alpha-1 antitrypsin. So emphysema is a chronic disorder in which the alveolar walls are damaged, due to which the respiratory surface is decreased. Okay, one of the major causes of this is cigarette smoking. Okay, main cause is cigarette smoking. What is going to happen? Alveolar walls are going to get damaged due to, and respiratory surface is going to decrease. Next one is occupational respiratory disorder. In certain industries, what happens, students, like grinding, okay, mine and all that, no, stone breaking, even this mill and everything, where you powder your wheat, rice and all of that, all those places where there's lot of accumulation of powders, okay, grinding or stone breaking, as much dust, okay, is produced, that the defense mechanism of the body cannot fully cope up, like to a limit. See, now I have dust allergy, that's why I have severe allergies in the morning. And it's also because the entire night, around like 7 hours or 6 hours, I am keeping the AC and the fan on, not both, any one of it. So now what happens is my body is not able to that 6 hours, it is tolerating, trying to cope up, and by the time I wake up, it will start reacting. But imagine it is in the case of a person who is working in an industry where there is so much dust every single day. Give me 1 minute. Okay, there is so much dust every single day that the body is not able to cope up. Okay, body is not able to cope up. So when body is not able to cope up, what is going to happen? They're going to have inflammations, they're going to have sneezing, which will lead to fibrosis, okay, where the lungs, okay, where the fibrous tissue will start proliferating in the entire lungs, causing lung damage, okay. And workers in such industry should wear protective mask, not the COVID mask that we wore, like proper shield kind of mask, which will not let even any dust to enter, and they'll have like small holes near the nostrils, which will not allow any kind of air to enter. Understood? So this is very important. Occupational respiratory disorders, that is in certain industries where there is grinding, okay, stone breaking, so much dust will be there that our body will not be able to cope up. So since our body cannot cope up, what is going to happen? Body is going to create symptoms, and it is going to grow the fibrous tissues. Okay, it's going to grow the fibrous tissues in the body, and this fibrous tissues is going to cause lung damage. And workers who are working there need to wear protective masks. And what happens, students? It usually happens, such kind of industries are placed in the very rural areas. So people there are not very aware, and at least the owners, or the, let's say, the people who are like the who, like, now I'm building the industry, I should be aware of it. I should be aware that, okay, if a person is exposed to this much dust every single day and for an hour, like 6-7 hours, then the person will have high chances of having breathing issues. So I should give them the protective mask, because these people are not going to have that knowledge. Okay, not everyone, I'll say, but some people do not have the knowledge, and that's why that mica industry, the entire issue came up during the COVID time, telling that we should stop using glitters in our cosmetics, because there are small kids who are going under the tunnel, like burrows, to take this mica. And what happens is, because it's being digged at a very wrong ratio of digging it somewhere down and up, what happens is as they're moving down, these stones will start falling on them, and many people lost their life. Okay, you can just check the mica story in YouTube. You will be able to find the COVID time story. Okay, you'll be able to find the COVID time story where, like, a lot of kids have lost their life, and where there were families with 4-5 kids, and right now they have only 1 kid or no kid. It is all because because street students, you cannot dig a tunnel which is huge for a big person to go. What they will do, they'll build small tunnels, and these kids used to go inside the tunnel. Okay, they do not know, their parents are telling go and collect and come. They used to go and come, and it used to be those glittery things, so they used to go inside, collect and come. What happens when they go inside, as it is very dark and oxygen also is less, some kids get breathing issue, and many people have died because of the the breaking down of the rocks. So it's very sad, and this people actually loot this urban area, people telling that you do this, I'll give you 400 rupees per day, or wage, per day wage I'll give you. And these people don't understand what could be the health issue, and later when they lose their kids, at one point they just become so stone hearted, they're like this is what we do for living, we do not have an option. So, like, at that point, so it's our responsibility that if we are building such industries, and we are the ones who are educated, and we are building industries in the rural area, it's our responsibility to make sure that these people are not harmed doing work for us, right? That's what we should. So students, yes, we have done our breathing and exchange. I hope all of you enjoyed the chapter. I'm so sorry that today the entire day I had so much of cold, and my rhinovirus attack was very extremely high. That's why in middle I had to go on and off, but we have completed the chapter, and I hope I have also made the concepts clear for you. So see you all in next class. Thank you so much for joining, and do not forget to like and also comment and let me know how you're enjoying the class.